Welcome to Up Close, the research, opinion and analysis podcast from the University of Melbourne, Australia.
I'm Shane Huntington. Thanks for joining us. In this episode, we'll be discussing stem cells, and in particular what are called induced pluripotent stem cells, or iPS cells. Because they're typically derived from adult somatic cells, iPS cells avoid some of the ethical controversies that surround the use of embryonic stem cells. To hear more about iPS cells, how they contrast with embryonic stem cells, and the promise they bring to treatment of heritable diseases, we are joined by Dr Mirella Dottori, head of the Stem Cell Laboratory within the Centre for Neuroscience here at the University of Melbourne, and Dr Alice Pébay, senior scientist and head of the Cardiac Differentiation Program of the Stem Cell Medicine Group at the O'Brien Institute. Welcome to Up Close, Mirella and Alice.
Thank you. Good morning.
Alice, let me start with you. For those of our listeners who are not aware, what is a stem cell and whereabouts do we find these in the body?
So there are different types of stem cells, in fact. We have to differentiate between what's called an embryonic stem cell and an adult stem cell. Let's start with embryonic stem cells. As the name suggests, an embryonic stem cell is a cell that comes from the embryo. Mm-hmm. It's called a stem cell because it means that it's a cell that is not really becoming anything yet. It has potency to become everything. An embryonic stem cells come from very early stages of an embryo, while an adult stem cell is a cell that is found in niches within our body, within an adult body or a children's body.
What role do these stem cells actually play? I mean, they obviously have a very specific function in the body. What is that role?
So an embryonic stem cell will, of course, have a fundamental role, which is to make an individual. Without an embryonic stem cells, we wouldn't have a body. An individual. They're here to make very early on in development the cells that will create the skin, the muscle, the different organs, the nervous system, all of the organs of our body. So if we go back to adult stem cells in the body, these adult stem cells have the potential to differentiate into specific cell types. They have less potential than embryonic stem cells, which means that they are probably in the body to generate cells of a specific part of the body. For instance, bone marrow stem cells will give rise to blood cells throughout our life, basically.
So embryonic stem cells can become essentially any cell, whereas the adult stem cells have a very specific function. Is that right?
Yes, that's a good generalization, yeah.
When we talk about embryonic stem cells, can these cells be used in anyone's body from a particular source, or does there have to be some sort of inherited link between the cells? Does it have to be a family member? Can you just take embryonic stem cells and use them to help any person?
So you wouldn't take embryonic stem cells and implant them into someone. First of all, because we know that embryonic stem cells, because they can become basically everything, can also become cancer. We don't know how to control the differentiation, especially in a body. If you inject stem cells, you take a major risk of creating a new disease in your body. The second thing is rejection. If you put anything coming from another individual into your own body, you will take the risk of having your body rejecting it. So you wouldn't do that.
Mirella, let's talk just for a moment about the controversy that has surrounded the use of these cell types, in particular the embryonic cell types. What is the basis of this controversy?
The basis is that embryonic stem cells are derived from a blastocyst. So at the point of fertilization, so you have an egg and a sperm, they come together to form a zygote. And then the zygote keeps dividing, and after 4 or 5 days, it becomes what we call a blastocyst. So that blastocyst, at that time point in nature, normally it's travelling down like the fallopian tubes, and it's at that time point where it actually implants into the uterus. So the laws basically throughout the world is that most embryonic stem cells need to be derived from a blastocyst, but the blastocyst was generated for the purpose of in vitro fertilization. So couples that have undergone IVF and have all these different embryos, some of which they immediately implant. If they don't want to immediately implant them, they can freeze them down. After a certain number of years, they have a choice to make, either destroy the embryos or implant them. And now there's a third option, that they can donate these embryos into research. And if they donate them, one of the areas of research is to derive embryonic stem cells. So the controversy that surrounds it is that this embryo has the potential to give rise to a new individual. So in actual fact, many people see it as if you're destroying life in order to save other lives.
Some of our listeners will remember we covered some of the issues around stem cells in episode 4. But one of the questions I have for you now, several years later, is do you believe that the controversy around embryonic stem cells has helped to drive these other areas of stem cell research like the one you're focused on?
Absolutely. It's a very, very tricky question to answer, this whole controversy surrounding embryonic stem cells. When does life begin? Are we taking away life? And that's based upon people's beliefs and understandings, and there's no right or wrong about that. And obviously some people feel extremely strongly about this, understandably, and rightly so. Perhaps that is one of the main driving force, also from pure scientific research point of view, that people are thinking, okay, we know that the potential of using embryonic stem cells is extremely, extremely valuable because they have the potential to become all cell types. And hence we now have a source of cells that we can drive them into different cell types for cell replacement therapy and so forth. So what's an alternative way to go about this? Now, one of the alternative ways was through cloning, somatic cell nuclear transfers. And for many, many years, people have been trying to go down that path. However, for humans, this is extremely inefficient. People have managed to clone to a certain point in the dish but haven't been able to propagate it. I'd just like to point out that in cloning, there's therapeutic cloning and reproductive cloning. Therapeutic cloning is that it's just for the purpose of deriving cell types, and reproductive cloning, which is against the law internationally, would be to implant that clone into a mother to derive a new individual. Now, because of that inefficiency, again, people have had to think about a third option, and for a number of years people have been looking at what are the essential genes or factors that make an embryonic stem cell that it's pluripotent, that it can become any cell type. And amazingly, some scientists from Japan and also in the US, but the Japanese scientist driven by Yamanaka was one of the leaders, the first groups, that were able to narrow it down to 4 or 5 factors fundamentally that were like the key driving genes that make an embryonic stem cell its pluripotent characteristics. From there, we were able to derive these induced pluripotent stem cells.
Mirella, we often hear about the value of cord blood these days, and in fact many parents are encouraged by private companies, by some hospitals, to store their blood, sometimes at high cost, sometimes donating their blood freely. What is the sort of purpose of this? What's the value in cord blood with regards to stem cells?
Cord blood in many ways has similar characteristics to sort of bone marrow stem cells, essentially like an adult stem cell in that they can definitely give rise to some lineages, for example, some blood cell types, even perhaps some bone, fat, and so forth. Now, there's some schools of thought out there, or some researchers that also claim that cord blood can also give rise to all cell types of the body, even nerves or muscle and so forth. For that reason, many people have thought, okay, cord blood might be another cell type that might be equivalent in certain ways to an embryonic stem cell. So there's been a lot of trials done where people have even transplanted cord blood and have seen some sort of improvement. But one thing that many people have noticed with cord blood stem cells is that they release a lot of supportive factors, trophic support. So part of their regenerative potential is not that they're replacing diseased cells, it's more that they're providing support or factors to promote the body to regenerate and replace its own cells. And I think it's more that mechanism which is quite therapeutic in their use rather than a cell replacement. Now, the idea of storing cord blood, I think it's a tricky one. Actually, a lot of my friends and family ask me that advice, simply because we don't know where the research is going. So in some ways you can see the value of having your own cells as close as possible stored for 30, 40 years down the track. That say if you get a leukemia, if you get some sort of condition, you've got the cells there as a source. But now again with the technology of iPS cells where we can derive potentially cells from the patient, maybe such things like storing your cord blood isn't as necessary as people thought. But why not store everything? So it's up to the individual. I think it's a tricky thing to answer, and I can understand both ways.
This is Up Close, coming to you from the University of Melbourne, Australia. I'm Shane Huntington, and our guests today are Dr Mirella Dottori and Dr Alice Pébay, and we're talking about stem cells. Alice, one of the notable aspects of embryonic stem cells is that they are pluripotent, whereas adult stem cells are often referred to as multipotent. Can you give us an idea of what the difference is here, what these two terms mean, and what gives rise to the various options that these cells give us?
I think it's a human construct, isn't it, where we need to categorize everything. So we've got multi, pluri, totipotent. Basically what it means is that if we take an embryonic stem cells, the potential of differentiations are almost for all cells. That's why it's called pluripotent. If you take an adult stem cells, it will have less differentiation potential, which is why it's called a multipotent stem cell. For instance, if I take a neural stem cell, this neural stem cell will be able to differentiate into cells of the nervous systems, such as astrocytes or macroglial cells in general, and neurons. If I take an embryonic stem cell, I will be able to differentiate this cell into different type of cell, cardiomyocytes, neurons, skin cells, much more potential.
Now, in your work, you start off with an adult skin cell, and you actually induce it to become a required cell type to do this work. Tell us a bit about what's happening there.
So what we did basically was following the research that was done in America and in Japan, which allowed reprogramming of an adult skin cell into a cell that resembled an embryonic stem cell. For this, we basically modified the expression of a few genes in skin cells that we obtained through biopsy, waited long enough, and eventually arrived to a stage where the cells looked like embryonic stem cells. Once they arrived at that stage, we were able, with our collaborators, to isolate these cells and then grow them in culture, in conditions of culture that are used for embryonic stem cell maintenance in the laboratory. So this way of reprogramming was by modifying genes and then using techniques that are normally used for embryonic stem cell biology.
When you say reprogramming, you're not quite making an embryonic stem cell here. So what's similar and what's different?
What's similar and what's different? When we know they are similar in the differentiation potentials, we know that they are similar in the expression of various what we call pluripotent markers, so markers that show that the cells are stem cells. But we know they are potentially different, and this is really very hot research today. But we know they are different in their epigenetics, and potentially they might be different in their ability to differentiate, but it's not sure yet. Basically, what seems to happen is that although iPS cells, which are the reprogrammed cells, are able to differentiate into various cell types, I certainly found in the laboratory that it's a bit harder than for human embryonic stem cells to do the same job. We have to push them a bit more to differentiate. Is it a technical problem? Is it a reprogramming problem? We don't know yet, and we're still working on this to understand.
Alice, what do you mean by epigenetics?
Well, a very simple explanation of epigenetic would be a modification that is not found in a gene that has the modification, but that you will find in the next generation. So it's epigenetics.
Alice, why did you choose skin cells for this work? What's specific about them?
So again, what we did with our work, which was to derive iPS cells from people with a specific genetic disease, we followed protocols that were already published by other scientists around the world. Why did they choose skin cells? There might be various reasons. One of them is the availability of skin cells. Skin is the biggest organ of the human body. It's very easy to get a skin biopsy and to reprogram it. There are, however, today a discussion about the best source of cells that should be used or that could be used for generation of iPS cells. And it seems, in fact, that the reprogramming efficacy might differ from one cell type to another. We've been using skin cells, but why not using adipocytes or fat cells or other cell types to derive iPS cells is totally open.
Mirella, we were talking earlier about some of the ethics involved around embryonic stem cell work. I can imagine one of the issues that would come up when you're doing this type of modification to a skin cell so that they can make many other cells is that, as mentioned earlier by Alice, embryonic stem cells allow you to essentially make the whole body.
Yes.
Does that become a problem with this work? Are you able to do the same sort of thing with your induced pluripotent stem cells?
In theory, yes. And actually they have shown that in the mouse, where they could take these iPS cells generated from mouse tails or skin cells and generated them into iPS cells, and then they implanted these iPS cells into another mouse blastocyst. Then they implanted the blastocyst into a pregnant mouse or pseudo-pregnant mouse. The progeny of that mouse is all derived from the original iPS cells. So in theory, it's almost like cloning. That procedure there is like cloning. So in theory, the potential is there that, yes, from an iPS cell, you can give rise to a whole new individual, and they've shown that.
It sounds almost like these cells are too good to be true.
Yes.
Are we looking at the sort of demolition of the embryonic stem cell field and this being replaced by this new type of induced cell that you're working on?
Absolutely not. It's not perfect. As Alice mentioned, there's several factors with iPS cell technology that still need to be addressed. And the fact that the way iPS cells are generated through essentially changing the whole genetic code, even that procedure is not perfect. So when people have looked at all of the genes that are expressed in embryonic stem cells and compared that with iPS cells, it's not a 100% perfect match. Because obviously we don't know everything. There's many things we're still not sure about. You wonder, well, where there isn't a perfect match, does that matter? And you never ever know. So in many ways, ES cells or embryonic stem cells are like the gold stamp. And we always need that to compare and contrast with iPS cells. Whether there's a need to keep deriving new ES cell lines, maybe, maybe not. Perhaps we don't need ES cells for therapies directly, but we always need them for research purposes.
Mirella, if you were to reach over right now and take a skin sample from me, and then you were to go through this process of inducing these cells to have these incredible characteristics, can you talk us through how you actually go about that in the lab, what the process is?
Okay, so you would go and see a doctor and would take out a small skin biopsy, leave one little stitch, if that. And then from your skin, we would basically culture the skin cells, similar to what they would do like from burn victims, where they're culturing the skin again to replace the skin cell, and expand them up in numbers. And then we introduce these 4 or 5 different genes or proteins, and there's different ways in which you can introduce these into cells. The traditional way and the most efficient way is by using viruses. And so by using viruses that carry these genes in there, they infect into the cells and they start expressing these genes, these 4 or 5 proteins that we call the Yamanaka factors. So it's OCT4, KLF4, c-MYC, and SOX2. So once these proteins or genes are expressed, they incredibly basically induce these skin cells to express these same stem cell genes. They're like keys, they're like drivers, and they're sort of like telling the cell, okay, we're here, so you start expressing all stem cell genes and silencing all the genes that make you a skin cell. So that's why it's called reprogramming. And then once these skin cells are tricked or converted or transformed or reprogrammed into becoming like a stem cell, they silence the genes that we introduce. Now, because the traditional way of using viruses obviously is not a therapeutically viable system, so now there's many groups, including our own lab, where we're using non-viral approaches. So you can either directly put in the protein or you use other sort of systems such as plasmids, which you can just bring these proteins into the cell. That's what's needed. You just need to bring these 4 or 5 proteins to make that switch. Once the switch has happened, you no longer need the exogenous genes.
What's the most challenging part of this particular process?
As incredible as it still is quite inefficient. So most groups around the world that do this, it's only about 0.1% of cells that actually end up being reprogrammed. So after we introduce these genes, we have to wait at least 3 weeks, and we wait patiently. We don't do anything. We pray to the right gods. After some time, you look down the microscope and suddenly you start seeing that these skin cells, which are normally long and elongated, they start changing their morphology and they become looking like stem cells, so they're much more smaller, rounder, and they start proliferating. And they form what we call colonies. Once we see that down the microscope, we think, that looks like they've reprogrammed. So we mechanically actually pick them, we isolate them, and we try and keep propagating them. If they stay propagated, then we know we're on the right track and starting to form a cell line. Then we do all the other characterizations that we have to look for other genes and see what they can do. Can they become nerves? Can they become muscle? Can they become this and that? Then we tick the box, yes, they're stem cells. But that whole process takes a while, and the most efficient sort of reports is still only 4%. So there's many, many challenges we still have to face. But the fact that it can be done is what everyone's excited about.
I'm Shane Huntington, and my guests today are Dr Mirella Dottori and Dr Alice Pébay. We're talking about stem cells here on Up Close, coming to you from the University of Melbourne, Australia. Alice, one of the areas of great interest that you're focusing on is the condition called Friedreich's ataxia. Can you give us an idea of what this is and what ataxia in particular is?
So Friedreich ataxia is a genetic disease, an autosomal recessive disease, which basically is touching a gene called frataxin. Basically, there is an expansion in the DNA of a triplet of a nucleotide. The expansion is too big and implies that the expression of the gene frataxin will not be done as in individuals who are not affected by Friedreich ataxia. What happens is that the gene is not expressed properly, which means that the protein frataxin is not found in sufficient number in the cell. This protein frataxin, although it's encoded by a nuclear gene, is migrating to the mitochondria. In the mitochondria, frataxin is involved in the transport of iron. There is an exchange of iron between the mitochondria and the cytoplasm. This modification ends up on the physiological level creating big issues, physiological issues for individuals, such as ataxia, loss of movement, tremor, loss of sensory neurons, loss of neurons in the cerebellum, loss, in fact, of many cell types. But that's the major problems that arise. There is also other problems, such as diabetes, that is linked to this disease, and problem with the heart. There is lots of heart problems, which are not one single type of heart problem. There's different type of heart problem. But generally, the fatalities are caused by heart problem in Friedreich ataxia.
Mirella, who does this actually affect, and at what age does it start?
So most commonly, it starts in childhood, perhaps late childhood, but there is also a late onset. So basically there is a direct correlation, as Alice mentioned, that the disease comes from a GAA, trinucleotide expansion within the frataxin gene.
Can you explain what you mean by this GAA expansion?
Basically, DNA is made up of 4 different types of nucleotides, G, A, T, C. Now, within the genome, sometimes you have these, what we call repeats of 2 nucleotides or 3 nucleotides that are similar, that are repeated in number. And sometimes, if there's too many of these repeats, it can cause disease, like Huntington's disease, for example. It's due to a specific repeat of these nucleotides expanded. In Friedreich's ataxia, a similar thing occurs. So they have specifically GAA, 2 of these nucleotides, but in trinucleotide clusters called GAA, they're expanded in numbers. So a normal individual may only have up to 20 GAA, whereas in Friedreich's ataxia, they've got about 500 GAAs, and so forth. Now, where this occurs is within the intronic regions of that gene. So that is not the regions of the gene that make the protein. It's the spaces in between, you could say. And because of that huge expansion, the DNA is very, very tightly coiled up. And so the rest of the proteins can't get access to it, or it can't open it up to have normal protein transcription occurring. So what happens is you have very low levels of the protein being expressed. So In Friedreich's ataxia, they don't get 0%. They only get like 9 or 10% being expressed. And therefore they get onset of symptoms. Because people who are carriers, if you've only got one chromosome that has this GAA repeat, they're fine. And they're carrying half the amount of protein. They're carrying 50%. Whereas in Friedreich's ataxia, they have both of these chromosomes, so they've inherited from both parents that have these GAA expansions. So from being instead of 100% or 50%, it's only 9 or 10%, which results in 9-10% of frataxin protein and hence the onset of symptoms and the disease characteristics. Now, they've been able to correlate that the longer the expansion, the earlier the onset of the disease symptoms and also the quicker the rate of the disease progression. So once you start getting symptoms of ataxia, loss of balance and so forth, it progresses pretty quickly where affected individuals within a few years need to be in a wheelchair. Their coordination is lost. And the type of nerves that tend to die are those that are involved in feeling and in weight and in balance. So it's not like motor neuron disease where they can't have muscle control. They do have muscle control, but they can't feel the ground underneath them or they can't feel things. And so because they don't have that feedback from the body, hence they can't stand up or they can't balance.
Now, when we talk about the use of induced pluripotent stem cells to treat this particular condition, how exactly will that work?
Okay, so there's several cell types that are affected in Friedreich's ataxia. The predominant ones are specific cell types within the nervous system that causes the ataxia, and in particular the heart cells. And it's the heart cells which eventually does kill them because they basically get severe heart problems. So with iPS cell technology, there's no clinical trials out there yet. It's too much in early stages. Even with using human pluripotent stem cells, such as embryonic stem cells, in terms of using them for cell replacement therapy, what is possible and what is very, very tricky. So replacing cell types of the heart is perhaps one day a more feasible option, even though still quite complex. Replacing cell types of the nervous system is incredibly complex, and most people think, it won't be possible, but who knows? You never know. Simply because it's like a wiring system, the circuitry. However, the possibility is still in there. So one possible thing would be to derive these iPS cells from patients, in an ideal world, correct the mutations, remove that GA expansion or introduce frataxin back in there in a gene in a proper state, which we can do in the lab, and then transform these iPS cells into the cell types that need to be replaced in the body. So that's one potential. The other very valuable use of these cells is that there's a huge lack of appropriate cell models of the disease because different cell types are affected, and the only cells that they've got to work with are mainly blood cells or skin cells from the patients, which are not really affected. But they're nevertheless isolating these cells from the patients and trying to do drug screening or understand disease pathogenesis. So now we have a source of, by using iPS cells from these patients, we can make iPS cells and drive them into the cell types that we know specifically degenerate in this disease, the heart cells and the nerve cells. Now that we've got the nerve cells and the heart cells of disease, we can perform drug screening assays to see that drugs will be able to increase frataxin levels in these cell types specifically. And that's really important. As you know, if you take a drug, not every cell responds in the same way. It's very specific to that cell type. And whilst there are some animal models of Friedreich's ataxia, they're quite limited. So having a cellular system would be highly useful.
Elise, let me ask you, with regards to the production of these cells and, growing them in the lab essentially, what's restricting that to this sort of low percentage success rate, and what are we doing to sort of get around that?
Yeah, in making iPS, the reprogramming technique is new. It's only 3-4 years ago that it was described, so many people around the world, including us, are working onto finding new techniques, non-viral, small molecule, a whole bunch of new techniques to improve the efficiency of reprogramming. But I think that's only one part of the research. Another important part as well, that once we get the iPS, even if it's low reprogramming, is to get an enrichment of the cells of interest. And that's another extremely important part of research, how to get from a small amount of cells a much higher amount of, in our case, cardiomyocytes or neurons. I think that's an essential question. And the complexity of this research, I guess, is also coming from the fact that, as we said before, iPS cells are similar to human embryonic stem cells. We do not know today how similar or how different really they are. So on top of studying a disease with iPS cells, we're still studying whether iPS cells are a very good model of development, for instance, like ES cells are, or if they have characteristics associated with the reprogramming. So this job is really complex at the moment because it's one, to study the disease, to look at drug screening, to try to get higher number of cells, but also to make sure that the cells we have generated are in fact a mirror or not of a human embryonic stem cell. So it makes it very complex.
Dr Mirella Dottori, head of the Stem Cell Laboratory within the Centre for Neuroscience here at the University of Melbourne, and Dr Alice Pébay, senior scientist and head of the Cardiac Differentiation Program of the Stem Cell Medicine Group at the O'Brien Institute. Thank you very much for being our guests on Up Close today and giving us an understanding of this exciting new area of research.
Thank you very much.
Thank you.
Relevant links, a full transcript and more info on this episode can be found at our website at upclose.unimelb.edu.au. Up Close is a production of the University of Melbourne, Australia. This episode was recorded on Thursday, the 24th of February, 2011. Our producers for this episode were Calvin Parham and Eric Van Bemmel. Audio engineering by Gavin Neighbour. Background research by Diani Lewis. Up Close is created by Eric Van Bemmel and Calvin Parham. I'm Shane Huntington. Until next time, goodbye.
You've been listening to Up Close. For more information, visit upclose.unimelb.edu.au. Copyright 2011, the University of Melbourne.